If I can figure it out, you too can figure it out. With simple motive, let's begin our discussion of hypersensitivity type 1 reactions. Okay, talking about hypersensitivity reaction, simply from the word it is clear, hyper and sensitivity. Hyper and sensitivity. Hyper means excessive and sensitivity simply means sensation. That means it is the condition because of hyper sensitivity of immune system. That's all. And we're talking about type 1 hypersensitivity reaction. It is also called allergy, or it has got several names, such as we can say it as immediate response, or also it can be said as allergy. Okay. There is one good definition given in Robbins. We'll be looking at definitions after completing our discussion about hypersensitivity type 1, because after completing only it will be so easier for us to understand that one. Okay, I've already mentioned that hypersensitivity is overactivation of immune system, because of which there will be a result of tissue damage, and it can be also said as immediate response or allergies, and we can say it as also anaphylactic type. What I said is anaphylactic type. And what is this anaphylactic type? Anaphylactic type is the condition of severe allergic condition. So now let's get toward the basic thing. Okay, before getting into this diagram, which was mentioned in Robbins 10th edition, I would like you to take you toward the pathogenicity. It is also the pathogenicity, but I would like you to take you toward the simpler form, through which we can have a general idea about what we are dealing right now. Okay, let us talk about the pathogenicity. Talking about the pathogenicity, let us draw one system, and this we have drawn while we are studying about the basic concept of immune activation. Okay, let us consider this is our environment and this is our human skin cells. Let us say this is our skin, intact skin, and our skin, or we can say it as a barrier, and this barrier or our intact skin is preventing these microorganisms or antigens to enter into our body. But because of some pathological condition, if our barrier get disturbed, or if there will be infection in our skin, then at that time these anatomical barrier will allow antigen to get into skin. Okay, and this will result. After that, what will happen is these skin contains various type of antigen presenting cell, and one of the example of antigen presenting cell is dendritic cells, and under that dendritic cells we have said that the dendritic cells found in skin is Langerhans cells. So let us say this is our APC, antigen presenting cells. This is our APC, antigen presenting cells. As antigen enters inside our skin, at the time the first thing that they will get exposed to is they will get phagocyzed by antigen presenting cell. As antigen enters inside the body, they get phagocyzed by antigen presenting cell. As antigen presenting cell phagocytes these antigens, they will disintegrate these antigen presenting cell into small peptides. Now let us draw here small peptides. They will disintegrate these antigen presenting cells into small peptide, and these antigen presenting cells, when divided into small peptides, they will be expressed into the surface of these APC with the help of what? With the help of MHC molecules, major histocompatibility complexes. Okay, with the help of this. Now everything it is happening beneath the skin. Now these APCs which are expressing this antigen in a form of small peptides on its surface with the help of MHC. What is this green one? This is MHC. Now they will go into circulation. They will go into circulation, and after going into circulation, as they reach lymphoid organ, and in lymphoid organ we can find a greater concentration of T cells, and in that T cells, while talking about that T cells, we have said that there are two type of T cells. One is helper T cell and another is cytotoxic T cell. Now here we can say these antigen presenting cells will present these antigen to what? Helper 2 type of cells, I mean TS2 cells. Okay, let me make it clear by drawing the diagram. Okay, and these antigen presenting cell, they are presenting here antigen. They are presenting antigen here to what? To helper 2 type of cell, and here in helper 2 type of cell we have got what? T cell receptors. Okay, T cell receptor. Okay, this is, and what we said is these here the type of helper cell that we are talking is TH2. What we said, it is TH2 cell, and we have already discussed about what type of cell will be activated. What type of cell will be activated? T helper cell. There are two type of T cells. Talking about T cells, we will recall the information that we discussed in T cells. There in T cells we are having T4 type, T helper 4 plus, CD4 type T cell, and one was CD4 and another was CD8 plus T cell. And we are saying this CD4 plus T cells will be activated when there will be MHC class 2 on the surface of what? APC. That means this one. This should be what? MHC class 2. And similarly, CD8 plus T cell will be activated when there will be MHC class 1 on the surface of what? Here. Okay, that means here we are saying helper type 2 cell are being activated. That means obviously this MHC was, what was this MHC? This MHC should be of which type? Type 2. And while talking about T helper cells, I have already mentioned that CD4 plus T cells are called helper cells, and these helper cells are also of several types. And we're talking about the different type of TH helper cells. There was TH1, TH2, and we had got TH13. And if you will remember the fact that we discussed in T cell lecture, then you will find out that TH1 cell was taking part in type 4 hypersensitivity reaction, and TH2 was taking part in type 1 hypersensitivity reaction, and type 13 was secreting interleukin 17 and was performing, and this interleukin 17 was taking part in antifungal and autoimmune disorders. Antifungal, and another function that it was performing was performing autoimmune disorder. Let's not say function, it was causing autoimmune disorders. And this was the thing that we studied in the type of CD4 T cells. It was the lecture on T cells. Okay, we have known about T helper type 2 cells. Okay, we have known a bit about T type 2 helper cells, T cells. Now as okay, whatever we have covered right now, we have said that these antigen will enter into our skin, and in its skin they will bind with APC, and as they will bind with APC, they will be disintegrated into smaller peptides, and as they will be disintegrated into small peptides, these will be expressed on the surface of MHC. And what is this MHC? It is class 2nd MHC. Why is this class 2nd MHC? Because we are saying that it will be activating T helper type 2 cell. That's all. Now these APC will be presenting antigen to T type 2 helper 2 cells. Now that means T type 2 helper cell will be activated. What will happen as T type 2 helper cell will be activated? After its activation, the thing that will happen is it will cause the production of interleukin 4 interleukin 5 and another interleukin 13. These are the things that will be secreted by activated type 2 helper T cell. That's all. Now, overall, if we will see overall the function of these things, then we will find out that these things will be increasing the concentration of eosinophils. Okay, what is the condition in which the concentration of eosinophil is increased called? The condition in which eosinophil concentration is increased is called eosinophilia. That means it will cause eosinophilia. It will cause the increase in concentration of eosinophils, that is called eosinophilia. Okay, as we are talking about eosinophilia, let us mention various conditions in which the concentration of eosinophils are increased. The various conditions in which the concentration of eosinophils are increased are, okay, we can write it right here. We can say the various conditions are, what are the various conditions? These eosinophils increase at the condition of allergies, and we can say in type 1 hypersensitivity reaction, also in the condition of parasitic infection. Okay, these are the conditions in which eosinophils are increased. Okay, now let us see the particular action of these things that are secreted by TS2 cells, that means interleukin 5. Okay, we're talking about interleukin 5. We'll find out. Okay, let me find something. Okay, we're talking about interleukin 5. Interleukin 5 performs one special functions. It causes the formation of antibody, IgE antibody. It causes the formation of, what it does is formation of IgE antibody. Okay, it causes the formation of IgE antibody. Now, as it forms IgE antibody, okay, as it forms IgE antibody, this IgE antibody, what it will do? It will go and it will attach with mast cells. Okay, attach with mast cells. Now, let us move toward mast cells. What are the mast cells? Okay, what are the mast cells? Mast cells are the derivatives of basophils that are found in bone marrow. Okay, let's say in normal condition. Okay, right now what we are discussing is, we are discussing the condition in which antigen was entering in our body, and we are discussing the condition in which antigen is entering in our body. Okay, let us suppose it is the normal condition. That means we are not talking about antigen entering in our body. At the normal condition, what we find is, we find mast cell. This is mast cells. What is mast cell having is, in normal condition, the mast cell is having receptor on its surface. These are the receptors on the surface of mast cells. What are these receptors for? These receptors are IgE antibody receptors. What I said is, these are IgE antibody receptors. At normal condition, there is the presence of receptor, and in this receptor at normal condition there is presence of nothing. But at the condition in which this antigen has entered in our body, the condition in which antigen has entered in our body, at that time there is the formation of IgE antibody. Now these IgE antibody will be going to mast cells, and in that mast cells they will be taking place here. Why they will be taking place here? Because obviously these are the site of IgE antibody receptor. Okay, that means IgE antibody will be attached here. Okay, as we know, we will draw antibody like this. How we'll draw antibody in a Y form like this. These are IgE antibody. Okay, this is mast cells, right? Inside mast cells, let us draw these structures. Okay, now here the condition that we are discussing is, we are discussing antigen entering in body for the first time. Okay, for the first time, look here, for the first time when antigen enters into the body, at that time eventually at the end there will be the production of what? IgE antibody. IgE antibody, and these antibody will be attached to the receptor present on mast cells. And this, the condition right now what we are speaking is up to this one. Okay, up to this one, whatever we have spoken up to this one, this is the condition of, this is condition, the condition in which for the first time antigen enters in our body. It is called the what? It is called the first exposure. What I said is first exposure. There are several names for first exposure. We can say first exposure as stage of sensitization, but we can say stage of sensitization, stage of sensitization, and it can be also called as priming, priming. And in this condition there won't be any symptom in body. That means no symptoms are visible at this condition. What we are saying is no symptoms, no symptoms visible, no symptoms are visible. Okay, now we have said it is the condition of first exposure. Now what? Okay, this is the condition of first exposure. Okay, let us see right here in the same diagram. We can see the figure of Robbins. Okay, these are the allergens. Allergens means we can say antigens, right? Allergens are the substance that will result in allergies, allergy condition. Okay, we can antigen, we can say it as antigen. We have mentioned is that antigen. This is the antigen. The antigen is in normal condition. This antigen cannot enter the surface, the surface that is mucosal lining, or we can say skin. This can't enter into skin, but at the condition when this barrier get ruptured, at that time they will enter into the skin, and into the skin they will, for the first time they will come in contact with APCs. We said that they will come in contact with APCs, and one example of APC is dendritic cells, and this is the dendritic cells. You can see right here, dendritic cells. That means these dendritic cell or APCs, they will engulf the antigen or allergen. And we can see another condition in which these antigens are also taken by B cells and they are expressed on the surface. Okay, that is also the condition. One condition, they are taken by dendritic cells, and these, by taking into dendritic cells, they will what? They will process, they will disintegrate these antigens and they will express into the surface. And after expressing into the surface, they go into the lymphoid organ, and into lymphoid organ we can find what? We can find a large amount of T cells, and they will present this antigen to T cells. And after presenting this antigen to T cells, we can see there is TS2 cell. TS2 cell, we'll see the TS2 cell being activated, TS2 cells. Okay, you can see here the B cell, it is also presenting these antigens to what? TS2 cells. Here the T cells that we are talking about is TS2 cells. Okay, after TS2 cell is activated, it will enhance B cells or antigen presenting cells to secrete what? To secrete antibody. And here the antibody that is produced is IgE. How? We said that the first antibody that is produced in primary immune response is IgM. How this IgE came? This came because of what? Switching, antibody switching that we have studied in B cell and T cell lecture. Okay, you can see here exposure to antigen or allergen, it is the same thing, and then happens activation of TS2 cell, and TS2 cell is activated and IgE class switching in B cell. That means IgE is formed by class switching. We have learned with the help of interleukin 4 and 5, right? We have learned, right? And after IgE is produced, that means due to IgE class switching, the production of IgE takes place. You can see a large number of IgE secreted by what? Plasma cells. Plasma cells are the activated form of what? B cells only. Okay, that's all. Now these IgE will move into mast cells, where they will go and attach. They will attach into the Fc receptor for what? IgE. These are the Fc. Fc is a component of what? This is the component of this. I mean, this is the part of what? Antibody. Fc is a part of antibody, right? And Fc is a part of antibody. So here the receptor will receive the Fc part of what? This IgE antibody. That's all. And this mast cell will be having what? Mast cell will be having mast cell will be having IgE antibody with it. Now what it will do is, here till now we were talking up to here, we talked up to here, that means up to this it was the condition of first exposure. And what we were telling the first exposure, we were telling the stage of sensitization, priming, and during this time there was no any symptom in body. So this was it. Now we will go toward the condition in which there will be repeated exposure. Okay, again the same antigen entering into body, again expose. Okay. When the same antigen will enter into the body, at that time what will happen is we will not go through all that process again, because we have already one what? We have already mast cell. And what this mast cell is having? This mast cell is having antibody on its surface. This is the mast cells. It is having antibody on its surface. We have already got this one. Okay, let me draw some other receptors. Okay, these are having antibody on the surface. Now again, when exposed to same antigen, these antigen, okay, let us show antigen with this. No, we have been showing with red, so we'll show with red only. So these antigen are coming from external environment. Now what they will do is they will cause cross reaction between these two antibodies. Now what they are doing on further exposure, this much was already present in our body. This was present at the time of what? At the time of first exposure. But again, when these antigen will come into our body, at that time what they will do is they will do what? What they will do? They will do cross linking, right? They will do cross linking. And what will happen when they will cause cross linking? Okay, let me write here what they will do. Antigen will do cross linking. Cross linking between what? Between antibody. Cross linking between antibody. Now this cross linking between antibody, what it will result into? It will result into mast cell activation. What I said is this will cause mast cell activation. And what will happen if these mast cells are activated? Now they will cause release of chemicals. Now what are these chemicals? These chemicals are, some are preformed, whereas some are freshly formed. What are the things that we will learn into preformed one? The thing that we learned into preformed one are histamines. And what are under the freshly formed one? Talking about the freshly formed are the one that are derived from we can say phospholipids, right, on the cell membrane. Okay, the freshly formed are arachnoic acid, the one that are formed when phospholipase is acted on the cell membrane. It will cause the formation of arachnoic acid, and these arachnoic acid will cause the formation of what? Leukotrienes through which cycle? Lipoxygenase, and they will also help in formation of what? They will help in formation of prostaglandins, PGS, and this process is what? Cyclooxygenase, or we can write COX, right? Okay, and these freshly formed, under freshly formed we have also got cytokines, right? One was arachnoic acid, another is cytokines. And talking about cytokines, what are the cytokines that are produced? The cytokines that are produced are, here comes interleukin 1, tumor necrotic factor, we can write interleukin 4, 5 and 13. Okay, and these, if you remember, these were the product that were produced when there was the activation of TS2 type of helper cells. You can see right here, where we can see right here 4, 5 and 13, right? Okay, now because of these things, I mean when these things, because of these things, histamines, leukotrienes, prostaglandins, interleukins 4, 5, 13, because of these things there arise a various type of complication, and that is what we are concerned about. That is what we are trying to prevent on this hypersensitivity reaction. Okay, now let us focus toward this one. Okay, before we have discussed till this one, I mean what it was doing is it was binding of IgE to Fc receptor on mast cells. Okay, this was the Fc receptor, right? Fc receptor obviously means that one receiving Fc. And what is the Fc? Fc is the portion of antibody, and what is this antibody? It is IgE. Okay, we completed this much. Now on repeat exposure to allergen, that means on repeat exposure to allergen, okay, again this allergen, it is again exposing, or it is again entering in our body. At that time what will happen? We had already, this one was already present into the body. Now this will come and this will cause cross-linking. What it will cause? It will cause cross-linking. It will cause what? Cross-linking. Cross-linking of antibodies. It will cause cross-linking of antibodies. As the cross-linking takes place, what will happen? Activation of mast cells. These mast cells will be activated, right? When these mast cells will be activated, they will cause what? They will cause the release of mediators. You can see right here, this is causing cross-link between this one, and the mast cells is activated. As it is activated, it is causing the release of several what? Mediators. Look, under the mediators, what we are talking about is, we are talking about vasoactive amines, lipid mediators, right, and cytokines. Okay, here it has divided it into this way, but here that time, I mean here where we are talking, we talked about we talked about preformed and freshly formed. The things are similar. Okay, now on vasoactive amines and lipid metabolites, we can see immediate hypersensitivity reactions, right, and a minute after repeated exposure to allergen. Okay, repeated exposure, this is the condition. These things will be produced on what? On being repeated exposure to allergens. And cytokines, where are these cytokines? What these cytokines are doing? They are playing role in late phase reaction. There is two type of reaction, we can say. One is immediate hypersensitivity reaction. Okay, the frequently just after repeated exposure to antigen, that is the immediate hypersensitivity reaction. And this is the late hypersensitivity reaction that will last for about 2 to 24 hours. We'll talk about this thing in where? We'll talk about this thing in this immediate reaction and late phase reaction. Don't worry, we'll make these things also clear. And this is what a brief idea, or briefly we talked about the pathogenicity of what? Of hypersensitivity reactions. Now let us talk here on detail about these, what these things do, right? What are the complications we'll face because of these release of these chemical mediators. Okay, let us talk about, okay, the complication that we'll find, that we'll see because of these chemical mediators are, we'll just write it right now. We'll list here. One is vasodilation. And this vasodilation is performed by what? It is performed by histamines and prostaglandins. Okay, histamines, and another is prostaglandins. And another complication that we'll face is bronchospasm, and this is the respiratory problem in lungs, right? Bronchospasm. And this bronchospasm, we'll see this bronchospasm also in what? Histamines. This will be caused by histamines, and we'll find that these are also caused by leukotrienes. Another complication we need to mention is itching and increase in inflammation. What I said, itching and inflammation increase, right? This is caused by what? This is also caused by, we can see that this is caused by histamines, and another one is cytokines. Okay, one important thing that we can see right here is histamine is common in all. That means histamine has a great role in causing different complications in hypersensitivity. Okay, now while talking about this thing, we can, okay, now as I said, as we mentioned that mast cells will be activated and there will be a release of different chemical mediators. Now let us see the diagram from 10th edition Robbins. Okay, here this is the mast cell that you are seeing. This is the mast cell, right? And here in the surface of mast cells we can see IgE specific Fc receptor for Fc portion of IgE. You can see here, right? You can see here IgE receptor for IgE specific Fc receptor, this one. Now this is receiving what? The Fc portion of IgE that you can see right here, right? This is IgE and it is being received by IgE specific Fc receptor, and it is the condition after repeated exposure of allergen. Now after repeated exposure of allergen, what this antigen does is this antigen causes cross linking between these two type of what? IgE antibody. As they cause the cross linking between two type of antibody, what will happen is this mast cell, it will get activated, right? It will get activated. This mast cell will get activated. As it gets activated, what will happen? What will happen is this will cause signal for cytokine gene activation, right? Cytokine gene activation. Cytokine gene activation, as it will cause the signal for cytokine gene activation. What will happen is this signal will be sent into nucleus, and this nucleus will secrete cytokines, right? And cytokines and chemokines. And what are these cytokines and chemokines? These are the one that will play important role in late phase. We haven't talked, in this diagram we haven't mentioned about early immediate phase. Okay, we are talking right now about late phase. That means these cytokines and chemokines will be participating in late phase reaction. And what are these cytokines and chemokines? Leukocyte infiltrations. What they will do is they will cause leukocyte infiltrations, epithelial damage, bronchospasm, right? And what are these cytokines? These leukotrienes B4, C4, D4. These are what? These are cytokines. Okay, we talked about signal for cytokines gene activation. Now another thing is there will be a signal for degranulation. That means the granular content that are present into mast cells that are released now because by the process of degranulations. And what are these granules content? These granule content are histamine and proteases, and we have already talked about their role in different complications. Okay, now another thing, important event that is happening is there will be the generation for signal of activation of phospholipase E2. What is this phospholipase E2? This phospholipase E2 will convert phospholipids in what? Okay, this will convert phospholipids in what? In cell membrane into what? Into arachnoic acid. And this arachnoic acid will what? Will be further forming. We have already talked. Okay, let us go here only. Look, you can see this was the signal. What was this? Signal for activation of phospholipase A2. Phospholipase A2 is enzyme that will cause the formation of what? Arachnoic acid. Okay, membrane phospholipid, it will form what? Arachnoic acid. Not only arachnoic acid, it will also call what? Platelet activating factor. Okay, we under arachnoic acid there was the formation of prostaglandin, and this process and the pathway that was forming prostaglandin was COX pathway, cyclooxygenase pathways, and there was formation of leukotrienes, and here was the lipoxygenase pathway. Okay, and what these things do is these things. Okay, here we talked about the late phase. Late phase there were in late phase reaction there was the role of cytokines and chemokines, and we are talking about immediate response, right? Immediate response. And in immediate response we can see these are other remaining things we have already studied here, right? Where we have studied is cytokines were helping in late phase reaction. Okay, if I haven't mentioned it earlier, then just keep in mind cytokines are responsible in late phase reaction. And what are the different event in late phase reaction? Leukotriene infiltration, epithelial damage, bronchospasm. And what are the thing going on in immediate responses? And the thing going on in immediate response is vasodilation, vascular leakage, smooth muscle spasm. And in immediate response we'll find what? Prostaglandins, right? We'll find what? Histamine, proteases, these things. Okay, This was all about the degranulation, I mean the role of mast cells in what? In hypersensitivity reaction part 1. Okay, now where we'll go? The thing that we'll discuss now is, as we are learning about the late phase and immediate response, okay, we'll see this diagram. What is this diagram all about? Okay, the first thing that I would like to make you clear is, this is the condition of repeated exposure. Repeated exposure, I mean in repeated exposure, when antigen will enter into body, okay, second time or third time, as it enter into the body, what are the things, what are the changes that we'll observe in immediate? What immediate means? It is immediate response means the response that we will see from the time of entry of antigen into body till 1 day. We can say about 1 day. You can see it is the entry and it is day 1. And what is the late phase reaction? The reaction taking place during the duration of 2 days, or we can say after 1 day to how many? After it was 0, I mean at the time of infection to first hour, then from second hour to 24 hours. This was the late phase reaction. I would like to make a correction. Immediate reaction. What was immediate reaction? Immediate reaction was the condition in which immediate reaction was the condition of exposure of antigen into the body to 1 hour of duration, and the late phase reaction was the condition from 2 hour to about 24 hours. Okay, what happens in immediate reaction? We have already said in immediate reaction what was going on there. In immediate reaction, as we can see in this figure, immediate reaction, vasodilation, vascular leakage, and smooth muscle spasm was going on. Okay, let me read out the thing that is written right here. Immediate reaction. The phase of immediate hypersensitivity reactions. Obviously, the figure 1, phase of immediate hypersensitivity reactions. Kinetic of immediate and late phase reactions are shown here. As we can see, the immediate vascular and smooth muscle reaction to allergen develop within minute after challenge. Okay, that means immediate reaction will take place after the minute of allergen entering into the body. We have already mentioned, right? Okay, and here the important thing that I have already said is, we are all talking about allergen exposure in previously sensitized individual. That means we are not talking about the first time one. The conversation is already over. The discussion that we have done first time, that means in which there were no any symptoms since. We have already finished that discussion. Okay. And the late phase reaction develop from 2 to 24 hours. We have already discussed about it. 2 to 24 hours, right? The immediate reaction B. Okay, you can see the diagram B. Okay, this is the diagram B. In immediate reaction, this is the diagram of immediate reaction. Immediate reaction. In immediate reaction, we can see there will be vasodilation. We have already said that there will be vasodilation, right? Vasodilation, congestion. What is this congestion? You can see vascular congestion. Vascular congestion simply means leakage of body fluid. That is what the congestion is. And edema, that causes edema, right? And we can see here, we can see edemas, right? And we can see, okay, edema. These things are the things that we will see at the time of immediate reaction, right? And we're talking about the late reaction. Okay, this one is the late reaction, diagram C. Here we can see that it is characterized by inflammatory infiltrate rich in eosinophils. You can see a large amount of eosinophils. Here we can see a lot of eosinophils. Okay, it is characterized by presence of inflammatory infiltrate rich in eosinophils. Okay, neutrophils also and T cells also. This was simply illustration of immediate reaction and late phase reaction. Okay, this was all about these things. Okay, now while proceeding forward, the thing that I would like to take you toward is, I would like to take you toward various example of disorder caused by immediate hypersensitivity reaction. And some of the example are, look here, anaphylaxis, which can be caused because of drugs, bee sting, or because of food, anything, right? And what will happen here? What are the pathological manifestation? Fall in blood pressure that may result in shock caused by vascular dilations, airway obstruction due to laryngeal edema, and we have already said that bronchial asthma, it can That means this is also a manifestation of what? Hypersensitivity type 1. And here the pathological manifestation are airway obstruction caused by bronchial smooth muscle hyperactivity, okay, inflammations, tissue injury caused by late phase reaction. Late phase reaction, you know what happens in late phase reaction. Similarly, there can be allergic rhinitis, sinusitis, hay fever, right, food allergies. And the pathological manifestation of allergical rhinitis, sinusitis are increased mucus secretion, inflammation of upper airway and sinuses, and food allergies, increased peristalsis due to constriction of intestinal mucosa. Okay. Now, as we have seen these examples, that means different allergies, right? Different allergies. In examples, we have talked about allergies, right? Now, let me illustrate in our one form allergies. Allergies. Talking about allergies, there can be allergy from food, there can be from allergies can be dermatitis, right, and rhinitis, right. Okay, these can be the allergy. Now, why these allergies takes place, right? Why they occur? Allergies. Why allergy occurs? Allergy occurs because of genetic makeup of individual. That means it is, we can say, if I have allergies, then my nephew or my grandfather or my father, they may also have allergies to certain type of stimulus, allergens, right? Okay. There is one important term that we need to know, and that is atopy. What is this atopy? Okay. This atopy, when genes are responsible for increasing incidence of allergy. That means if there is role of gene in increasing or in manifestation of allergies, then this condition is known as atopy, right? And various type of atopy that we can see is atopic dermatitis, atopic rhinitis. Let me mention. Atopic dermatitis, and atopic rhinitis. Okay. And as I have already said that if genes are responsible for causing allergy, then that condition is known as atopy. And we have seen some of the research have been done, and it was found that the reason for development of allergy, I mean the gene that are responsible for development of allergy in individual are mainly found in chromosome number 6. And if we recall the information about, if we recall the discussion that we made in MHC, then we can say that MHC are the major histocompatibility complex, and they are also found in chromosome number 6. So we can say that atopic condition and MHC, they are interrelated with each other. That means we can say that atopic, the gene responsible for causing atopy or for causing allergies, are present in chromosome number 6, and MHC are also present in chromosome number 6. Then we can build a relation about MHC and allergies or atopies. And that was the example of hypersensitivity reactions. And now talking further, we can see that there are also several drugs that are responsible for causing allergies. So in that condition, the most popular drugs, antibiotics, and among the antibiotics we can find penicillin. Penicillin is considered as the most notorious drug. So before administrating any of the penicillin to patient, we should take a proper history of patient if they are hypersensitive toward penicillin or not. And that is one of the important thing that we should consider before administrating penicillin, because it is if the person is having any of the allergy to penicillin and we are administrating him that, then there can be a condition of anaphylactic shock, so the patient may die. So it is very important to do that. Okay, now the key concept that we have discussed in this topic in hypersensitivity type 1 reaction are right? This is the immediate type 1 hypersensitivity. They are also called allergic reaction or allergies. They are introduced by environmental antigens, allergens. Obviously, the antigens that was entering in our body was from environment. That stimulates what? TS2 response. T helper type 2 cells they were stimulating them, and stimulation of type 2 helper cells was causing what? IgE production, right? What these IgE will do, this IgE will attach to mast cells by binding to Fce receptors, right? And this was on re-exposure to allergen, level of cross-linking of IgE and FcRI causes what? Activation of mast cells. Okay, IgE was attached with mast cells by binding to Fce receptor. On re-exposure, this was the condition of first exposure we have already discussed, right? On re-exposure to allergens, right, this will cause what? Cross-linking of IgE and FcRI receptor, and that causes the activation of mast cells and release of mediators. We have already learned about these things. The principal mediators are histamine, proteases, and other granular content prostaglandins, leukotrienes, and cytokines. Yes, we have already studied about these things. The mediators are responsible for immediate vascular and smooth muscle reactions. Yes, late phase reactions are responsible for causing inflammation, right? The clinical manifestation may be local or systemic and range from mildly annoying rhinitis to fatal anaphylaxis. So these were the things that we needed to discuss on the topic of what? On the topic of hypersensitivity. So this was all. Now the next lecture will be on hypersensitivity part type 2 reaction. So this much. Hope to see you again. Thank you.